gymnosperms are heterosporous, which means that they produce two types of spores, microspores and megaspores. Microspores are produced in microsporangia and megaspores are produced in megasporangia. Sporophylls bearing microsporangia are called microsporophylls and sporophylls bearing megasporangia are called megasporophylls. Microsporangia are seen on the lower side of the microsporophyll. Here is a microsporophyll of the Cycas tree. What you see here is actually the lower side of the microsporophyll with microsporangia. Let's have a closer look at the microsporangia. Oh, these microsporangia are all open and they have released their contents already. It's okay. Here is a megasporophyll of Cycas. And you can see the megasporangia. Inside the megasporangia, megaspores are produced. But the megasporangia in gymnosperms are different from that of Selaginella. In gymnosperms, the megasporangia are covered with a few protective layers called integuments. These megasporangia covered with integuments are called ovules. These ovules will become seeds later. You may remember that in Selaginella, microsporophylls bearing microsporangia and megasporophylls bearing megasporangia were seen in the same strobilus. But in gymnosperms, they are seen in separate strobili. The strobili in gymnosperms are also called as cones. So, the strobili or cones bearing microsporophylls and microsporangia are called microsporangiate strobili or male strobili or male cones. The strobili or cones bearing megasporophylls and megasporangia are called megasporangiate strobili or female strobili or female cones. The male and female strobili or cones may be present on the same tree as in the case of Pinus. Here, the female cone is bigger in size than that of the male cone. If you check a longitudinal section of the male and female cones of Pinus, you can see that the cones have a central axis around which microsporophylls or megasporophylls are arranged. The male and female cones may also be seen on different trees, as in the case of Cycas. But in Cycas, the megasporophylls are not arranged around a central axis in a cone. Instead, they are loosely arranged at the stem apex that appears like a rosette. Sometimes it may look like a cone, but it is not a true cone. So, a true female cone is absent in Cycas. Cycas plants have the largest ovules of the plant kingdom. So, let's see the structure of a Cycas ovule. The body of the ovule consisting of many cells is called nucellus. In other words, nucellus is the central part of an ovule. It is covered with 3 layers of protective layers called integuments. There is an opening called micropyle and a small chamber called the pollen chamber. Here is the close-up look of micropyle and pollen chamber. I will explain about these structures later. Now, let's check the microsporangia. Inside the microsporangia, the haploid microspores will germinate into male gametophytes called pollen grains. These pollen grains will later produce the male gametes or sperms. Each pollen grain consists of 3 cells, a prothallel cell, a generative cell and a tube cell. At this 3-cell stage, the pollen grains are released from the microsporangia. They are carried away by wind. In this video, what you see like a smoke is actually the pollen dispersal. Check out the link of this video in the description box below. This kind of massive pollen dispersal occurs during the spring season. Sometimes this causes allergic rhinitis or hay fever. The pollen grains carried away by wind will get trapped in a sticky drop called the pollination drop, seen at the micropyle. Pollination drop is secreted by the ovules. This transfer of pollen from the male part of a plant to the female part of the same plant or another plant is called pollination. In almost all gymnosperms, as we saw in the video, wind plays an important role in the pollination. Such a kind of pollination by wind is called anemophily. Here, the pollen grains can travel with wind for miles and can withstand extreme drying. Now, the pollination drop dries out and the pollen grains are drawn into the pollen chamber of the ovule. The micropyle is closed. A tube called pollen tube starts to develop from the pollen grain. After a month of pollination, one cell of the nucellus gets differentiated into a megaspore mother cell. The megaspore mother cell undergoes meiosis division to form 4 haploid megaspores. Among the 4 haploid megaspores, only one becomes functional. The other 3 megaspores will degenerate, in other words, dies off. After a long period of time, nearly 1 year, the functional megaspore undergoes mitosis division to form a female gametophyte. This female gametophyte is also called endosperm. Female gametophyte will produce female gamete or egg in the female sex organs called archegonia. Archegonia are seen at the micropylar end of the ovule. Sometimes 2 to 8 archegonia are formed by the female gametophyte. By this time, in the pollen grain which is in the pollen chamber of the ovule, the generative cell divides into a stalk cell and a body cell. Finally, the body cell divides to form 2 sperms. As you might have already noted, in gymnosperms, male sex organs or antheridia are not formed. The pollen tube carrying the male gametes or sperms grows towards the archegonia in the ovules. If the sperms are ciliated like in Cycas and Ginkgo, the pollen tube will discharge the sperms near the mouth of the archegonia and the sperms will move to the egg. But some gymnosperms like Pinus and Gnetum have sperms with no flagella or cilia. They move along the pollen tube to reach the egg inside the archegonia. The sperms and eggs of the Cycas are the largest in the plant kingdom. Out of the 2 sperms, one fuses with the egg and the other sperm degenerates. In the same way, sperms from different pollen grains will fertilize the eggs in each archegonia. In this picture, I have shown the case of just one pollen grain. The sperm nucleus and the egg nucleus fuse to form a diploid zygote. In the case of algae, bryophytes and pteridophytes, there was a requirement of water for the sperms in the antheridia to reach egg in the archegonia. But in gymnosperms, one thing to be noted is that there was no requirement of water for the fertilization to occur. Pollen grains reached the ovule by wind and the sperms reached the egg with the help of the pollen tube. The zygote now secretes a cell wall and becomes an oospore. The oospore then develops into a young embryo inside the ovule. Since a single Cycas ovule contains 2 to 8 archegonia, 2 to 8 embryos will also develop inside a single ovule. The phenomenon of development of more than one embryo inside a single ovule is known as polyembryony. Among the 2 to 8 embryos, only one embryo will reach maturity, others degenerate. All these changes that occurred after fertilization make the ovule turn into a seed. So, seed is basically a fertilized ovule. Seeds have many advantages. They are protected with many layers of cells. They can be dispersed by wind, water or animals. They can remain dormant till the conditions are suitable for germination. The food stored inside, that is the endosperm, can be used up by the embryo at the time of germination. When conditions are favorable for germination, the seed will germinate into a young diploid sporophyte, which is the main plant body of the gymnosperms.